So before I get to the chapter, I want to mention that these activities, these experiments, you cannot really just ignore them because you might need them for your SPM paper 3, or it might also come out in the subjective questions. They will ask you, number one, maybe how to set up the experiment, and number two, what are the results from the experiment. But normally, if they come out in subjective, they will ask you how to set it up. So you don't have to know this now because there's so many of them. There's not enough time to understand all of them. But just remember that when you're revising, don't ignore these experiments. I'd recommend you to buy a reference book that has answers to these activities because we can't do all of these experiments and the textbook does not give answers. So let's get into the chapter. 2.1 talks about cells, which are basically the basic unit of all living things. So this is the most important thing in the chapter, the components of animal and plant cells and their functions. Firstly, mitochondria are the powerhouse of the cell. What that means is they are the place where energy is produced. So energy is produced through cellular respiration, and energy is released in the form of ATP. The centriole, or some might call it the centrosome, I'll just write here centrosome, is what helps form the spindle fibers that you see during mitosis and meiosis, cell division. We'll know more about them in the other chapters. Next is the Golgi Apparatus, and the Golgi Apparatus is one of the more complicated ones, in my opinion. But the main thing that you have to know is the word here modifies. So if you see the word modify in objective, it's definitely Golgi, because no other example has modifying. But in case of subjective, you have to memorize the whole function. Processes, modifies, packs, and transports chemicals such as protein, carbohydrate, and glycoprotein. So how do you identify Golgi from the endoplasmic reticulum? Normally, you would see this flattened sacs like this, and then you see these sacs at the end. It's hard to explain, but when you see like the forecast papers, how they look like, you will be able to identify them a bit easier, I guess. Next is the plasma membrane. We normally call them the cell membrane, but SPM prefers the word plasma membrane. And the main thing that you have to know is that they are made of proteins and phospholipids. I think we'll get more to this later. But the main thing that you have to know is that, number one, it separates the cell from the external environment, and number two, it controls the movement of substances into and out of the cell, which causes the allowance of exchange of nutrients, respiratory gases, and waste materials between cells and the external surroundings. Oops. Next is the lysosome, and the lysosome is basically the doctor cell, the cleanup cell, as I like to call it, the cleanup cell. It contains these hydrolytic enzymes, which hydrolyzes complex organic molecules such as protein, nucleic acid, and lipids. And this helps to break down bacteria and components of damaged cells. So basically, it kills off the damaged cells so that they do not harm the body further. Next is nucleus, or nuclei for plural. And this is the most important thing in cells. Without a nucleus, the cell cannot function, because it controls all of the cell activities. Basically, all of the important things are here, such as the chromosomes, and nucleolus. Next is the ribosome, and the ribosomes consist of protein and RNA. And basically, ribosomes are sites for protein synthesis. Next is the endoplasmic reticulum, and it looks similar to the Golgi body, where it is a lot of flattened sacs. But as I said, the Golgi apparatus looks a bit bigger. I don't know how to say that, but you will see that the Golgi apparatus is a bit more specific, is a bit more to it than the endoplasmic reticulum. There are two main types, rough and smooth. Rough endoplasmic reticulum have ribosomes, smooth do not. So rough ones transport proteins synthesized by ribosomes, while uh, while smooth ones synthesize and transport glycerol and lipids and carries out the detoxification of drugs and metabolic byproducts. So basically, the endoplasmic reticulum is where a lot of things are transported, as well as the Golgi apparatus. I don't know if they mentioned the whole process in the book. I hope not, because there's a lot to know. Next will be the plant cells. So firstly is the vacuole, and the vacuole is a liquid-filled sac. And the main thing that you have to know is that animal cells might have vacuole. Actually, wait, I forgot if they do have vacuole, but basically, plant cells have very obvious vacuoles. Animal cells don't really do. And its main function is to determine the turgidity of the cell. When the vacuole absorbs water, the cell becomes turgid. Next is the chloroplast, and the chloroplast contains chlorophyll, and I'm sure everyone knows what the chloroplast does now. Next is the cytoplasm. I don't know why they included it here, because it's in both examples, but basically it is the medium for these cell reactions to happen. Like the cell has a nucleus and then a membrane. Between the nucleus and the membrane is all of these these small, I forgot what they're called, these components, these cell components. But for these cell components to even work together, there needs to be a cytoplasm. And the cell wall is very rigid. It is made out of cellulose. It is fully permeable, and the main thing that it does is to maintain the shape of plant cells, which helps with mechanical support. So these are the similarities and differences. The similarities is that they both have nucleus, cytoplasm, plasma membrane, Golgi, mitochondria, endoplasmic reticulum, and ribosomes. The differences are plant cells are fixed, while animal cells are not. And this is because they don't have a cell wall. Animal cells don't have a cell wall. Next, plant cells have chloroplasts, and they have a very large vacuole. And plant cells store carbohydrates in the form of starch, but animal cells store it in the form of glycogen. And animal cells have centrioles, but plant cells do not.